All right, having been introduced to a number of the big concepts in biology, now we'll take an introductory tour to the parts of the cell. Here we see two pictures, two figures of the same cell. I'll start on the right. You can see that this large area that's in blue, this is referred to as the cytosol. This is all the material that's inside the plasma membrane, but not inside an organelle. Lots of things occur inside the cytosol, and we'll learn about those. Now going to the left side, these are the various parts that we will study and learn about in the class. The nucleus, the mitochondria, the Golgi apparatus, the endoplasmic reticulum, and so on. This is a slightly more detailed, more realistic picture of a cell, and I will use this several times as we discuss different parts of the cell in succession. I will occasionally refer to comparisons between a city and a cell, comparing the functions that a city needs to do and a cell needs to perform. This analogy was created by Tim Gartner, the person who taught the course before I did, and I find it to be a very useful analogy. Let's start by talking about the nucleus here, this double membrane enclosed area. This is where the DNA is kept, where the information is stored. For a city, this might be a department of records or the archives of the city. You can also notice a portion of the nucleus, this stippled dark gray area. That's a portion called the nucleolus. This is an area of specific, very high activity. It's involved with the manufacture of ribosomes, the protein machines that make new proteins when they get out into the cytosol. So this is an area of very high activity, but it's part of the nucleus though. Here we see an electron micrograph where the DNA is stored. Here are the boundaries of the nucleus. Here are the boundaries of a nucleolus. Nucleoli are usually a single object or space rather than the two areas you see here. The nucleolus might be extending beyond or underneath the plane that the electron micrograph was taken in, a slice through a three-dimensional object. Not only is the information stored here in the nucleus by the DNA, this is where DNA is converted into an RNA molecule, when a gene gets turned on. This is utilizing part of the information to achieve a particular action. The RNA molecule that is made will then get transported out into the cytosol, where it will be used to manufacture protein. The power plant of a cell is a mitochondrion, and there are many mitochondria per cell. I'll highlight in this one. You can see that there's compartmentalization within the mitochondrion. There's an outer membrane, which encloses a white space, and there's an inner mitochondrial membrane, which is enclosing sort of a gold or tan space. Just to highlight their numbers, here's a fluorescence micrograph of a cell. We have one nucleus in a cell, but we have hundreds to a few thousands of mitochondria. Being the power plant of the cell, this is where ATP is produced. We have glucose breakdown products entering the mitochondria and being utilized to generate a high-energy molecule, ATP, which then can be used to power many energetically costly syntheses or functions elsewhere in the cell. Here's an electron micrograph of a mitochondria. I'll start by pointing out that we have a double membrane structure with an outer mitochondrial membrane and an inner mitochondrial membrane enclosing this area, referred to as the inner mitochondrial matrix. Here is a figure which is intended to correlate closely with the electron micrograph. And an important thing to notice from the original cartoon is that the outer mitochondrial membrane and the inner mitochondrial membrane are very close to each other. The inner mitochondrial membrane will extend all the way almost across the cell so that the inner membrane space is very small compared to the mitochondrial matrix, which is all the region inside here. The reason you have so much inner mitochondrial membrane is that most of the energy-producing process occurs by proteins that are sitting in the inner mitochondrial membrane. And molecules that are in the inner membrane space will cross through the inner mitochondrial membrane into the matrix. As the molecules get transported, we will capture some of their potential energy in the form of ATP, which then can diffuse out into the cell and power the various biochemical reactions that are necessary for cell growth. Now, protein synthesis can occur in two places. They can occur in ribosomes here in the cytosol, these dots, or they can occur in ribosomes on the endoplasmic reticulum. Proteins that are made in the endoplasmic reticulum are destined for an organelle or cellular membrane, or to be secreted to the external environment. Ribosomes in the cytosol are for proteins that are free in the cytosol or can get transported into other locations like the nucleus. As we have already mentioned, the entire protein synthesis process starts in the nucleus, where DNA gets converted to RNA. The RNA will diffuse out into the cytosol, be transported either to the rough ER or the ribosomes in the cytosol. Here we see an electron micrograph, and you can see the ribosomes dotting the endoplasmic reticulum, which is a membrane-based vesicle. In this dotted appearance from the ribosomes, that refers to this portion of the endoplasmic reticulum as the rough endoplasmic reticulum. There's also an organelle called the smooth endoplasmic reticulum, which we won't talk a lot about in the course. That's a site of lipid synthesis in the cell. The Golgi apparatus is sometimes simply referred to as the Golgi, is a series of stacked membranes that are involved in protein sorting. We have proteins being synthesized in the rough ER. These will migrate to the Golgi, and first the Golgi will process the protein a little bit. They'll attach things like sugars and other post-translational modifications. They'll also do a little quality control to make sure the protein is folded properly. And then it will package the protein into vesicles and sort the protein. Maybe the proteins need to go to the plasma membrane. Maybe the proteins need to be secreted to the extracellular environment. This all occurs in the Golgi. The plasma membrane is what separates outside from inside. It is the border of the city, or the border of the cell. But it's not only a barrier, it's also in charge of import and export of molecules that are outside the cell that the cell wants to take advantage of. It'll import those molecules into the cell, and it will also help to secrete things. On this slide, we see the way that the plasma membrane does that. If there are molecules in high concentration in the outside world that the cell would like to use, they will likely collect on receptor proteins sitting here in the plasma membrane. They will be concentrated in a particular area. The membrane will invaginate, and then it will be pinched off, creating a vesicle, which will then fuse with other organelles inside the cell for processing. Quite frequently, the processing involves proteases, chewing up proteins, nucleases, chewing up nucleic acids into building blocks for the cell to use to make its own proteins and own nucleic acids. This process is called endocytosis, and we'll learn more about this in the second part of the course. Conversely, the export process is called exocytosis, where we have molecules being concentrated in a portion of the endoplasmic reticulum, forming a vesicle with the contents inside it. Like soap bubbles colliding, the vesicles can collide with the membrane. They will then fuse, and the vesicles will open up. The contents of the vesicle will be released into the environment, and the curvature stress of the vesicle will be relieved and will tend to flatten out over time, creating some additional plasma membrane. Cells need a transport and structural scaffolding system. There are three parts highlighted here: microtubule and actin, which are involved in both transport and structure, and then intermediate filaments, which are less involved with transport but are primarily a structural component. Let's talk first about microtubules. These are the roads or the highways of the cell. They typically extend radially from a centrosome, and if something is produced near the center of the cell that needs to get to the outside of the cell, it doesn't want to rely on diffusion. A motor protein will attach to the cargo and essentially walk the cargo out along the microtubules to the periphery of the cell. Actin filaments. You've heard of actin and myosin in muscles. An actin is a molecule which can produce motion by polymerization and depolymerization. In this picture, you might imagine that the cell is moving in this direction. Actin is in many places inside a cell, but can also be concentrated at the leading edge of cell movement. The intermediate filaments, in contrast to microtubules, are mostly structural, and they are mostly circumferential around the cell. These provide rigidity and mechanical strength to the cell structure. These will all be talked about in the second part of the course. The cell needs a waste processing plant. The cell has two different types, primarily peroxisomes and lysosomes, and they have slightly different functions. Lysosomes are mostly an enzymatic waste processing plant. Proteases and nucleases will degrade proteins and nucleic acids in there into building blocks that the cell can use. The peroxisome is a chemical processing plant. Inside the peroxisome, there's lots of aggressive reactive molecules, primarily oxygen radicals like hydrogen peroxide, which is where organic molecules encountered in the environment, referred to as xenobiotic molecules, will be transported. Examples of xenobiotics include many of the compounds we've learned to fear: plasticizer-type molecules, drugs, other chemical compounds that the cell is not equipped to deal with. Many of these will be degraded chemically in the peroxisome and then the parts disposed of. In this slide, we've highlighted again the nucleus, which is involved with information storage with DNA. And we've also talked about its role in transcription, where DNA gets turned into RNA. This is also the region where the regulatory networks, the control center of the cell, are. It's the place where external information is integrated and the proper response is determined by figuring out what genes to turn on in response to a new environmental clue. And the way environmental clues get into the nucleus is by signaling pathways. This is the communication system of the cell. They typically will start with a protein on the plasma membrane binding an environmental molecule, which gives it a clue. This information will be transported via a protein relay to the center of the cell, often ultimately turning proteins on or off by phosphorylating them. The information is eventually transported into the nucleus, where transcription factor might be phosphorylated, and the activated transcription factor will transcribe some genes for eventual protein synthesis in response to a new environmental stimuli. So that will conclude our whirlwind tour of the cell. I'll tell you by the end of the course, you'll know each of these parts intimately. And in the next set of lectures, we'll talk about the molecular building blocks of the cell. Thank you.